end of today, biologists will be able to predict ratios of complete inheritance by practicing problems. We're going to do a lot of practice problems today, so I want you all to stay with me. I want you all to be engaged. I want you all to be participatory in the chat and taking yourselves off of mute so that you can give me some answers. The essential question that we seek to answer by the end of today's lesson: why do we use Punnett squares to predict offspring ratios? Why do we use Punnett squares to predict offspring ratios? So you all are finishing up writing today's title, which is Inheritance Part 1, writing today's date, which is October 27th, 2020, and writing Unit 7, Day 2. We do this to practice organizational skills. I want you all to be able to go back into your notes to be able to find exactly what you're looking for. If you're looking for information that was Unit 4, Day 3, you should be able to flip back easily in your notes and find that information. I will be moving on in 3, 2, 1. So we introduced the concept of inheritance yesterday. We defined inheritance as the process by which genetic material is passed on from one generation to the next. And as we often do, we think in terms of humans. We think from human beings, from parents to their children. But we also have to acknowledge that all living things need to be able to pass on their genetic material. It doesn't matter if we are talking about E. coli, which is a specific strain of bacteria. It doesn't matter if we are talking about the pollen and that the plants use to fertilize each other outside. It doesn't matter if we're talking about a bird species. It doesn't matter if we're talking about an insect species. It doesn't matter if we're talking about a polar marine species. Everybody needs to be able to pass on their genetic information. And when we think about this process of inheritance, we've also got to acknowledge that a portion of it is totally random. And in this process of fertilization, when the egg meets the sperm, or when the sperm meets the egg, I should say, there's no way of really knowing what the outcome of that fertilization will be. We don't have any way of knowing what the specific genetic makeup of the offspring will be. And that's because this is a random process. But in order to make educated guesses about what some of the potential traits of the offspring will be, we use these Punnett squares. Punnett. We use Punnett squares to determine the potential ratios of offspring genotypes and phenotypes. Definitely be writing that definition down, but as you write, can I have someone take themselves off of mute and tell me what the difference between a genotype and a phenotype is? What is the difference between a genotype and a phenotype? We've got 24 brilliantly bright young people on this call. You all are in a great position to answer this question because most of you were here yesterday. So what's the difference between genotype and phenotype? Phenotype is what you can physically see, and genotype is all about the genes. Thank you, Alyssa. So a phenotype describes physical characteristics of something. Oftentimes, these are the things that you can see. You can see skin color, hair texture, eye color, height, weight, there are also some things that you maybe can't see, but that can still be described by phenotype. For example, the shape of a blood cell. Some people have sickle-shaped blood cells. Of course, is not good. Most people have more bowl-shaped red blood cells. So even those descriptions would be considered phenotypes because they are physical characteristics. A genotype is a description of the genes, just like Alyssa said. Okay, and so yesterday we talked about the fact that everybody has different alleles. Alleles. You get one allele from your mom, one allele from your dad. And an allele is just a copy of a gene. Okay, let's move on. Oh, no, let me point this out. So we're looking here at a Punnett square. It looks like a 4-space chart. On the outside of the Punnett square, we've got the genotype of the parents. In this case, we've got the father on the top because these are the sperm cells, and the mother on the left side because these are egg cells. So actually, let's take a step back because I want to really make sure you all understand this. All right. So let's say that I've got 3 genotypes here. I've got A, I've got B, and I've got C. Nope, don't want to use that. Use G. Can you all see my screen?
Yeah.
Okay, thank you. So what would this genotype be called? We introduced 3 terms yesterday: homozygous dominant, heterozygous, and homozygous recessive.
Bro, Mark. Don't make me get on your cake. Hey, what's up, Miss Red? Look. Look, Mark, look.
Anthony, do you know the answer?
Of course not.
Okay, well,
I'm trying to
let's
figure
make
it
sure
out.
let's get some help from one of your classmates.
What was the question?
Yesterday we introduced 3 terms: homozygous dominant, heterozygous, and homozygous recessive.
Oh, the
So
first one,
this,
homozygous dominant.
excellent. So I'm going to write HD.
The second one is homozygous recessive.
The
No,
second one?
heterozygous.
Yes, good.
And the third one is homozygous recessive.
Good. All right, let me move down a little bit here. Now, some of us weren't here on the call yesterday, so they might be a little bit confused. So what we're saying is homozygous. Kyrie, what does the prefix homo mean?
Opposite.
No,
You
not
know?
opposite. It means the same.
All right.
So in this case, we have 2 homozygous genotypes. We've got one that is homozygous dominant and one that is homozygous recessive. What's the difference between dominant and recessive? Anyone can answer
Dominant
that.
is like you gonna have that trait because like it's like the strongest gene, and like
Excellent. Yeah,
recessive,
that's correct.
it's a possibility that you could have it.
Right. So good. So when we have a dominant trait like we have over here, we use capital letters. So the dominant allele is capitalized. And then our recessive allele is lowercase. But there's something else. We don't just have homozygous dominant and heterozygous. We can also have something else. And Kyrie told us we can also have something called heterozygous. What does the prefix hetero mean?
Different.
Excellent. Hetero means different. So in this case, we've got different sized letters. We've got a capital letter and a lowercase letter. So that means it has to be heterozygous because hetero means different. Now, each of these is an example of what we call, zoom out, oops. Each of these is an example of what we call a genotype. Okay, so when you see two letters like this, AA, BB, GG, what we're talking about is a genotype. And as Alyssa told us earlier, that means that we're referring to the genes. Each of these letters is called an allele. It's a copy of a gene. So let me write that down and then we'll get ready to go back to the. So each of these letters, for example, this lowercase g right here is called an A. Oops, that's ugly. It's called an ALLELE, an allele, which is a copy of a gene. So if I put two alleles together, then I have a genotype. So if you weren't present for yesterday's call, then it might be a good idea to try to write this down. We've got three potential genotypes: homozygous dominant, which we could say HD, heterozygous, which we could say HE, and homozygous recessive, which is HR. Homozygous dominant has two capitalized alleles, two big letters. Heterozygous has a capital letter and a lowercase letter. And homozygous recessive has two lowercase letters. And when I say letters, what I'm talking about is an allele. Every letter is an allele, a copy of a gene. Does this make sense? Any questions about this? Are there any questions about this? Now was definitely the time to ask. Oh, I didn't see your answers in there, you guys. I apologize, but thank you, Jessica and Alyssa and Makira. Any questions about anything that's on the screen right now?
No.
Thank you. Okay. All right. So let's come back over here. So we've got to be familiar with that lingo. We've got to be familiar with that lingo. Genotype, allele, homozygous recessive, homozygous dominant, heterozygous. We've got to know what we mean when these terms come up. So a Punnett square allows us to make predictions about the phenotype and the genotype of an offspring. Here's how you can solve a Punnett square. I want you guys to write these steps down. I know they're not highlighted, but I want you definitely need to have these steps written down. We will be referring back to the steps. The first step is to make a key. You need to know what each of the alleles represents and make a key. And again, when we say an allele, we're talking about a letter. So you've got to make a key. Once you've made your key, you've also got to determine what the parental genotypes are. So what are the genotypes of the parents? Are they homozygous dominant? Are they homozygous recessive? Is one heterozygous and the other is homozygous dominant? You got to figure out what the genotypes of the parents are. Typically, that information will be given to you. Once you've figured that out, you're going to write that on the outside of the box. You're going to write that on the outside of the box. So you could put dad on top and mom on the left, or you could put mom on top and dad on the left. But you're going to write the parental genotypes on the outside of the box. Once you've done that, you can start to do the actual cross, meaning that you start to combine the alleles. So as we look at this animation that's on the screen right now, we can see that the T's from the top genotype get dropped down into each of the boxes. And the lowercase red T's from the left, homozygous recessive genotype, move over into each of the boxes. And then lastly, you should be able to determine the ratios of the genotypes and phenotypes, or answer whatever question is being asked. So we're going to do a lot of practice with this today. So now that you've written down the steps, someone tell me, what's the first step that we need to do?
Turn the cells yellow.
No. Did you write down the steps from the last slide? What's the first step that we just saw on the last slide?
I'm gonna read it.
It says green leaves are dominant to yellow leaves, but what's our first step? See, Jessica says determine. Nick says determine the alleles. Okay, let's go back for a second because it seems okay. Determine what each allele means and make your key. So Nick and Jessica, you are basically right, but I'm looking for. We need to make a key. That's our first step. So in this case, we're going to use the letter G. In this example, we will use the letter G. A capital G is going to obviously represent the dominant allele. We always use capital letters for the dominant allele. And so that means that the capital G represents green. A lowercase g is going to represent yellow. It's the recessive allele. What's our second step now?
Split the two genotypes.
Excellent. So now we need to know the parental genotypes so that we can split them. So let's look and see what they tell us. So in step 2, they're telling us that mom's genotype is lowercase g. What do we call this genotype with 2 lowercase letters? What's this called? When I have 2 lowercase letters, what is that called? Yes, thank you, Nick. 2 lowercase letters is called homozygous recessive. So mom's genotype is homozygous recessive. And what about dad's genotype? If I've got a capital G and a lowercase g, I've got one of each. It's a hybrid. What do I call this? If I have one of each. Thank you, Jessica. It's called heterozygous, HE. We can call that HE. So I've got a homozygous recessive parent and I've got a heterozygous parent. Now we need to combine them. So I'm going to come back over here so I can show you what this looks like. First, let me make my actual Punnett square. So I'm going to draw, and this is something that you all should definitely be writing in your notes. Whoa, I don't know why that happened. Okay, hold on. So I'm just going to make my Punnett square. I'm going to draw my 4 squares inside of it. You should definitely be writing this in your notes. This is very helpful. On the last slide, we found out that mom is heterozygous recessive. That means that mom's leaves are yellow and she's got 2 lowercase letters. Dad is heterozygous. So dad has green leaves, even though he does have one one of his alleles is recessive. One of his alleles is lowercase. But anytime we've got the heterozygous genotype, it still expresses the dominant trait. Let me say that one more time. Anytime I have a heterozygous genotype, meaning one capital and one lowercase, it will still express the dominant trait. So this dominant allele is going to take over the smaller allele. So both HD and HE express a dominant trait. The only time you can get a recessive trait is if you have homozygous recessive. The only time I get a recessive trait is when I have homozygous recessive. All right. So let's go back to our Punnett square. So they're telling us now that I've got two parents. We've got our genotypes already written out. How do I want to write them? Should I put mom on top? Let's put mom on top. So mom's got two alleles. She's got a lowercase g that I'm going to put right there. She's also got another lowercase g, which I'm going to put right there. On the left side, I'm going to write dad's genotype. He's got a capital G, a dominant G, dominant allele. So I'm going to write that there, but he also has a recessive allele. So I'm going to write this right there. So now what I have is mom on top and dad on the left. My next step is then to do the actual cross. So I'm going to actually write these genotypes in my boxes, in my Punnett square. So I'm going to take this G and I'm going to put it right here. And I'm going to take this G and I'm going to put it right here. So I end up with G, G in this box. And then again, I'm going to take this G and I'm going to put it over here. I'm going to take this G and put it right here. So I end up again with G, G. Now down in the bottom row of my Punnett square, dad has a recessive allele. He's got a lowercase g. So I'm going to put this G here and this G here. So that gives me two lowercase g's in this case. And then take this G and put it over here. Take this G and put it over here. So again, two lowercase g's. So now we've got a lot of arrows. Hopefully you were following me along the way. But what you're seeing is that we just do the actual cross. I take my G's and I distribute them in the boxes. I take my alleles and I just distribute them in the boxes. Now, inside of the boxes, I have one. Let me just put this out too, just so it's very clear to see. We've got 1, 2. Oops, that's ugly. 2, 3, 4, I've got 4 offspring inside of my boxes. Let's go back to the slides and make sure that we did everything correctly. So they put mom on top just like we did. They put dad on the left just like we did. Dad's got a heterozygous genotype, so 1 green allele and 1 yellow allele. And then they did the crosses. Our last step is to analyze the results. So let me go back over here now. So how many green offspring do I have? And how many yellow offspring do I have? How many green offspring do I have? And how many yellow offspring do I have? Hello? Are we confused about something? Someone ask a question if we're confused. Yes, we're looking for the phenotype, so green or yellow. So remember, phenotype and genotype are just 2 different ways of describing the same thing. So let's go back up. So my genotype might be it might be homozygous dominant, like this one. But my phenotype is going to be whatever the dominant trait is. In our case, the dominant trait is green leaves. The recessive trait is yellow leaves. So the only way that I can get the recessive trait is if I have a genotype that is homozygous recessive. So genotype and phenotype are 2 sides of the same coin. They describe the same thing in different ways. But now that I've done my Punnett square, I've got 4 different genotypes here. I've got capital G, lowercase g in offspring 1. I've got capital G, lowercase g for offspring 2. I've got 2 lowercase g's for offspring 3 and 2 lowercase g's for offspring 4. So I've got 4 genotypes. All I'm asking now is for you all to tell me the phenotypes. How many of these offspring are green and how many of the offspring are yellow? Don't be discouraged if you're a little confused. This can be difficult stuff the first time. Thank you, Fanique. I have 2 green offspring. And so how many yellow offspring do I have? There are only 4 offspring. 1, 2, 3, 4. There are only 4 of them. So I think what you did, Fanique, hold on. That's 6. Okay, so here's the thing, guys. This whole thing is 1 offspring. Remember, we each need 2 copies of the same gene. So 1 organism has 2 alleles, 2 letters. So that's 1 offspring. This is 1 offspring. This is 1 offspring. This is 1 offspring. So I only have 4 total offspring. So there's no way I can have 6 yellow, especially if I have 2 green. If 2 of them are green, then 2 have to be yellow. Let's take another look at this. Offspring number 1, which is in the top left box, has a dominant allele and a recessive allele. So because it has different alleles, 1 dominant and 1 recessive, offspring number 1 is what we call heterozygous. It's different. So it's a heterozygous offspring. Anytime you've got a heterozygous offspring, that offspring is going to express the dominant trait. So it becomes green. Same thing with offspring number 2. It's heterozygous. It has a dominant capital allele and a recessive lowercase allele. So that means that it too expresses the dominant trait. It is also green. Offspring number 3, though, does not have a dominant allele. It just has 2 recessive alleles, 2 lowercase alleles. And we call that homozygous recessive. They are the same size, they're both lowercase. So homo means same size, but they are lowercase, so it's recessive. So that means that this expresses the recessive trait, which in this case is yellow. And the same thing for offspring number 4. So my result is that I have 2 green offspring and 2 yellow offspring. So if these 2 leaves mate, what we're saying is that 2 out of 4 of their offspring are green, or 50%. And 2 out of 4 are yellow, which is also 50%. So there's a 50-50 chance that their offspring will be green or yellow. So this is important stuff, folks. This is important. We're going to continue to practice with it. I know it can be tricky at first, but I definitely need to have some more participation. I'm only hearing right now from, Nick, Jessica, and Fanique. I heard from Kyrie a little bit too, and Alyssa a little bit. But a lot of you need to start chipping in so I can understand where your deficiencies are. It's okay to be wrong. It's okay to be confused. There's absolutely nothing wrong with that. What's wrong is when you all are silent the entire time and I have no idea how I can help you understand something. All right, so I like people who ask questions. That means that you're paying attention and you're trying to get better. So let's come back to the slides. So we ended up with the right results. 2 green and 2 yellow. That's the phenotypic ratio. The genotypic ratio means we have 2 heterozygous offspring and 2 homozygous recessive offspring. Okay, we're not going to worry about this right now. Let's focus on this. All right. All right, let's do more. I'm going to do another Punnett square here. Again, this should all be going into your notes.
First two is capital A.
What'd you say?
It's capital. They both.
Okay, let me see. Yeah. All right, so in this case, I've got one parent on the top that is heterozygous, meaning 1 capital letter and 1 lowercase letter. And then I've got another parent on the left that is homozygous dominant. So same size letters, and they are the dominant letters. So they're the capital letters. So 2 capital A's. Now I need to do my crossing. So this A is going to go here and here. This A is going to go here and here. This A is going to go here and here. And this A is going to go here and here. So maybe the arrows with different colors will help you all. But let's see. I also want to keep in mind that I've got, as is the case of these Punnett squares, 4 different boxes for 4 different offspring. So what's going to be the genotype? What's going to be the genotype in box number 1? What's the genotype in box number 1? Thank you, Alyssa. I should have 2 capital A's. We call that HD, homozygous dominant. What about in box number 2? What's my genotype there? Thank you, Jessica. Thank you, Fanique. 8, I've got a capital A and a lowercase a. We call that HE. That's heterozygous. What about in box 3? What's my phenotype? I'm sorry, my genotype. Yep, thanks, Jessica. Thank you, Fanique. I should have A, 2 capital A's. Again, that's homozygous dominant, HD. And then what about in box 4? Thank you, Jessica. So capital A and the lowercase a. So let's just keep in mind something here. H homozygous, homodominant equals AA. hetero equals AA. Homo recessive equals AA. So how many homozygous dominant offspring are there in this Punnett square? How many capital A are there? Thank you, Fanique. Excellent. I've got 2 homozygous dominant. What about hetero? How many hetero offspring do I have? Thank you, Jessica. I also have 2 of those. How many homo recessive do I have? Lowercase, lowercase. I have 0 of those, none of those in my Punnett square. I don't see any lowercase, lowercase in this Punnett square. So no homo recessive. Excellent. So let's come back to the slides and make sure that we got that correct. A, homozygous dominant, uppercase, lowercase, that's hetero, uppercase, uppercase, that's homo dominant, uppercase, lowercase, that's hetero. So Jessica told us we have 220, and they were correct. Jessica and Fanique, thank you so much. All right. So in the last one, we figured out the genotypic ratio. We figured out our how many of each genotype we had. Now we want to focus not on the genotype, but on the phenotype. We're told that we've got a key. The capital B represents brown hair. The lowercase b represents blonde hair. So we've got that key. So that's the first step. We've got our parents already set up. That's the second step. So now we just need to actually perform our cross. BB, uppercase, lowercase, uppercase, lowercase, uppercase, lowercase, and uppercase, lowercase. Notice how I had one parent that was homo dominant, and I had another parent that was homo recessive. And so all children are going to end up hetero. All of their children will have a heterozygous genotype. But what is the phenotype of the children? How many brown-haired offspring will I have? How many brown-haired offspring do I get from this cross? Excellent. Thank you, Alyssa, Jessica, and Fanique. So everybody else, I hope you are staying with us and taking notes. I should have 4 offspring with brown hair. Oh, wow. The reason for that is that a heterozygous genotype still expresses the dominant trait. A heterozygous genotype still expresses the dominant trait. This is what I was talking about right here. Homozygous dominant and heterozygous express the dominant trait. We were already told that the dominant trait in this case is brown hair. If all of my children are heterozygous, then they should all have brown hair. I shouldn't have any children with blonde hair. Any offspring, no offspring should have blonde hair. Let's keep going. Okay, let me come back over here. Now, we've seen that when we are given the parents, we can find the genotypes of the children. Can we also go backwards? Oops, that's real bad. So this time, I'm giving you the genotypes of the offspring, and I want you to give me the genotypes of the parents. How can I do that? If I've already got what's inside of the box, can I figure out what is on the outside of the box? Yes, I can do that. All I need to do really is reverse the arrows. These arrows that we did last time, I really just need to reverse them and take them the opposite direction. And I only need to do it for 2 boxes. So let's see what I mean by that. This G had to have come from right here. So that means that there has to be a G here. This G had to have come right here. So there has to be a capital G here. Likewise, this G had to have come from right here, that lowercase g. So there's got to be a lowercase g right there. And this G over here had to have come from up here. There has to be a lowercase g there. So do you see how I literally just reverse my arrows and I figure out what's on the outside of the box? You should also note that I didn't even really need to pay attention to these boxes. I only needed to focus on 2 of them, that box and that box. And now I know my parental genotype. Parent 1 equals heterozygous. Parent 2 equals also heterozygous. So if you know what's inside of the Punnett square, then you should also be able to figure out what's on the outside. This is going to take more practice, but we'll get there. So what we've been talking about today is called complete inheritance. And you should definitely write down this definition, please. Complete inheritance. We call it complete because the dominant allele, the capital letter, will always mask the recessive allele, the lowercase letter. The dominant allele masks the recessive allele. This means that you either have the trait or you don't. There is no in between. You have it or you don't. This also means that, as we've talked about, a heterozygous genotype will result in the dominant phenotype. A heterozygous genotype will show the dominant phenotype. Okay. So let's finish up with some practice, some additional practice. It says brown eyes are dominant to blue eyes. If a heterozygous brown-eyed man, heterozygous, so I've got one capital and one lowercase, they're different. If a heterozygous brown-eyed man is married to a blue-eyed woman, she's homozygous recessive, two lowercase letters, what are the chances they will make a blue-eyed baby? All right, so we need to do a cross between this heterozygous brown-eyed man and a blue-eyed woman. So step one is to make your key. The capital B, of course, represents brown. The lowercase b, as it says in the first sentence of the question, represents blue. My parental genotypes, it says that the woman, the mom, is lowercase b, so she's homozygous recessive. The dad is capital B, lowercase b, so he's heterozygous. Step three, we're going to combine the alleles. So I'm going to write my dad on top, capital B, lowercase b. There should also be a lowercase b here for mom. It's going to come up in a couple of clicks. Now I'm going to do my cross. So this capital B goes in this box, and it gets paired with this lowercase b. So I end up with a heterozygous offspring. Right here, this lowercase b goes into this box, and so does this lowercase b. So I end up with a homo-recessive genotype in this box. Now this capital B gets dropped down into this box, and this lowercase b gets pushed over into this box, so I still have another heterozygous. This lowercase b drops down into this box. This lowercase b gets pushed over into this box. So I end up with two lowercase b's. That's called homozygous recessive. And there it is right there. So how many of these offspring are blue-eyed? How many blue-eyed offspring are there in that cross, in that Punnett square? Thank you, Fanique. I've got two blue-eyed offspring. So what are the chances? What's the percentage that my offspring, or that the offspring of these two people, will be blue-eyed? If there are 2 out of 4, then what's the percentage? %50. Thank you. I've got 2 out of 4. 2 divided by 4 is 0.5, which is 50%. 2 out of 4 will have the blue eyes. The only way for me to get a blue-eyed baby is with homozygous recessive. So if these two people decide to have a child together, there's a 50% chance that that child will have blue eyes. There's also a 50% chance that they have brown eyes. All right, so right-handedness is dominant to left-handedness. We want to cross a right-handed individual and a left-handed individual. What percentage of their offspring will write with their left hand? Okay, so let's really write this one out. This will be the last one we do. You all do still have an exit ticket and an asynchronous assignment for the day. So please don't forget about that. I'm going to send a remind message to you all to go out at about 4 o'clock. as a reminder. So someone give me a letter. What letter do we want to use in this example for our right-handedness and left-handedness? What letter should we use? Could be anything. It doesn't matter. All right. Thank you, Makira. So we're going to use the letter R. So a capital R should represent what? If right-handedness is dominant to left-handedness, then what does the capital R represent? If right-handedness is dominant to left-handedness, then what does the capital R represent? Come on, folks, let's finish strong. We only have a little over 3 minutes left. Let's get this question done. I'll say it one more time. If right-handedness is dominant to left-handedness, then what does my capital R represent?
Your right hand.
Thank you, Joseph. So that means that R represents right-handedness. I'm just going to write the word right. And then a lowercase r should represent what? Thank you, Jessica. Left. All right. So it says we've got parent 1 is homozygous right-handed. Parent 1 is homozygous right-handed. So what should their genotype be? And thank you, Nick and Fanique, as well. If they're homozygous right-handed, what should their genotype be? Homozygous right-handed. I'm looking for, so if we've got a homozygous right-handed parent, what should their genotype be? We know it's homozygous, so we know that they have to be the same sized letters. Should it be 2 capital R's or 2 lowercase r's? Okay, so Nick says homozygous recessive, but we already know that this person is right-handed, and we know that right-handedness is dominant to left-handedness. So it's not homorecessive, it's homodominant, which means that should I use the capital or the lowercase?
Lowercase.
No. It's dominant, so I should use the capital letters.
Oh, I thought you said recessive.
Okay, let's pick up on this question tomorrow because you guys really need to fully understand this concept before we can move on. We only have 1 minute left. Please do take care of the exit ticket. There is an asynchronous assignment for the day, but if you're not fully understanding these concepts, then it's going to be difficult to do. So I want to pick up here tomorrow. Yes, I appreciate the 5 of you who have been consistently participating. Everyone else, we need to see more from you. Okay, if you're a little lost, it's good to ask questions. I like questions. I also